So good morning, everyone. Thank you for the invitation to speak at the school. My name is Adam Smelko. I am a postdoctoral researcher at Barcelona Supercomputing Center, or BSC for short. And today I'll be talking about a few stuff, but mainly agent-based modeling tool called PhysiCell. I'm not really used to 90 minutes presentations, so it may be the case that it will be as exhausting for me as it will be for you. And also a big disclaimer, although I am here at the life track and at BSc I work at Life Sciences department, my main specialization is in high performance computing and GPU acceleration. So it may happen that if you ask some biologically related questions, I will not be able to answer you. But I will try, so have patience with me. Okay. So let me start by talking a little bit about BSc. So institutionally, BSc is a consortium. It is funded by Spanish and Catalan government and also by University UPC in Barcelona. And it is hosting more than 1,400 scientists among different research fields, from computer science, earth science, to life science. And I will talk about this many times in the presentation, but I think that the interdisciplinarity is very important, mainly in BSc, and also in the topic that I will present today, because agent-based modeling tool lives in an intersection between math, biology, and large-scale computing. Yeah, so BSc is part of few groups. One of them is the Spanish RES group, which contains 14 nodes, and each of these nodes provides some HPC HPC resources. Also, BSc is part of Euro HPC joint undertaking, which is the European scale group, where the guests, the parts there are other well-known names such as Lumi, Leonardo, or Jupiter. And of course, in the heart of BSc, there is Mare Nostrum 5, the current flagship supercomputer. This supercomputer is no homogeneous at all. It supports the traditional MPI-based computing, but also GPU-accelerated or AI-accelerated workloads. And the new addition to Mare Nostrum 5 is actually the quantum partition called ONA. It is quite a humble one. As of today, we are supporting only 5 qubits, if I'm correct. But being able to SSH to quantum computer and send jobs there is indeed very cool. Okay, so in parallel to classical HPC, Europe is also investing heavily in these things called AI factories. And AI factories are supposed to provide the whole environment from training to deploying AI. And it should be accessible to both academia and industry. So yeah, these factories provide large-scale compute, curated data sets, domain experts, everything that you would need to train and deploy your AI, which is quite interesting. However, further in the European roadmap, there is ambition to create something much bigger, which is called gigafactories. And these are supposed to be ultra-large-scale infrastructures with at least 100,000 GPUs, so that we can, as a Europe, train foundational frontier models that are somehow in maintenance of a public governance, and they are scientifically open. So this is a great big shift, right? Because instead of relying exclusively on some commercial providers, Europe is investing in some sovereign and open AI, which is always nice. So this is one of the examples. BSC together with Spain is creating this foundational language model that should support the Spanish and other co-official languages. And these models are open. They span from 7 billion to 14 million parameters, and you can download them today from Hugging Face if you want. And yeah, this is just one of other highlight that I want to show, which is the use of this foundational model for something more scientific. So this is Project Carmen, where the AI is tuned to extract or structure typically unstructured clinical data. Yeah, so on the right you can see that we have some symptoms, some synonyms that AI can extract from the clinical text. So let me switch the topic a little bit and let me talk about this concept that perhaps is the one that unites all of the BSC fields, and this is digital twins. And digital twins is not a project or a discipline. It is a way of thinking of simulation and prediction together. So the idea is quite simple, but still also quite ambitious. You want to somehow create a computational representation of a very complex system, such as Earth, a human, or something like a digital chip, and make this representation not only descriptive, but in a way predictive and actionable, so that you can create actionable results from the simulation or a prediction that this digital twin of a real thing made. Yeah, and perhaps the most major example of this would be the Earth system. I believe that you saw this few days ago, I think yesterday, yeah. So climate and Earth system is it's been developed for decades, but today we can reach kilometer scale of global simulations. And there are initiatives such as Destination Earth, which if we have enough of compute power and smart enough models, we can simulate processes across the entire planet. And yeah, these models, we cannot say they are completely perfect, but we can already do some informed policy or disaster preparedness or long-term planning based on these predictions. So now the natural question arises. Now we have these amazing models, so why not a digital twin of a human? And this is a great slide. So here you can see a real factory. Actually, on the left part, you can see a real factory. It is a factory of a Siemens, I think. And on the right, you can see its whole digital twin. The Siemens cooperated with NVIDIA, with this Omniverse project. Maybe you heard about it. It is their physical AI. And yeah, there is the digital twin of a factory that can do some predictions, do some optimizations there without the need of trying it real life, trying to tune the factory manually, which is tedious and takes some time. So let's follow this thinking process and let's apply it to the human. So imagine that a patient would come to the hospital, and instead of carrying some treatment on the patient would be created a digital twin. So let's imagine that there would be some reference human model which would be parameterized by the personalized data of the patient, and then this model would be treated multiple treatments. And here is where the digital representative of the real thing shines. You can actually do multiple treatments and also in a shorter amount of time, because you can simulate much faster than our clock stick. Yeah, and now imagine that you would somehow evaluate this treatment, pick the best one, and then apply it to the real patient. So when will we have digital twins? Will it be next year, next decade, next week. The thing is that it is quite difficult. So, unlike Earth, where we relatively know the governing equations there, the whole human body is composed of many biological system interactions that span many orders of magnitude, both in space and in time. So, yeah, while this idea is super compelling, we must be honest and we must say that it is much more difficult problem, both scientifically in way of finding the correct models and methods, and also computationally to have enough computational power to simulate the whole human. But the silver lining here is that, yeah, we do not have to wait for the perfect solution. Instead, we can already build partial or task-specific digital twin. So one concrete example that I will be talking about more today is the cancer modeling. So here we can already connect some patient-specific data, such as genomics, single-cell sequencing data, to mechanistic computational models of a tumor. And yeah, these are inherently multi-scale. They link intracellular signaling, cell movement, cell behavior, up to the whole population level dynamics. Yeah. So while this is not a complete digital twin, we can already have some digital twin of a specific disease process, and according to that we can act, and it is also grounded on the real patient data. Yeah, so we can already ask some relevant practical questions such as therapy design and drug response. So let me talk a little more about this example. So one of our group at BSC simulated this petri dish of AGS cancer cells, and they let it simulate for some time, and at one specific point in time, a drug would be administered. And there would be actually multiple different drugs. And each cell had underneath its signaling network composed from Boolean network. And if this Boolean network was set up in a way that if a drug was administered, some signaling pathway would be triggered so that the cell population would either stop growing and dividing or continue. And yeah, on the top, you have the graphs of how the population of the cells was growing. And for this, we had some experimental data that we can base our simulation on, and that is the blue line. And the dotted line is our simulation results. So indeed, in this first step, we needed to tune our simulation so that we can match the experimental baseline. And this indeed required to run multiple simulations, where in each simulation we needed to turn the knobs of the simulation parameters so that we can well, perfectly match the result. And you can see that we reached a reasonable correlation. But then what we did was that we tried to simulate something that our models did not see before, did not ever see before, and that is the combination of the drugs that was administered. Yeah. So when and as you can see on the bottom graphs, the combinations of drugs, our simulation of how the population grows when the combination of drugs was administered correlates relatively well with the experimental data. So yeah, from this we could already do some predictions that otherwise we could not do. So what is the next other thing that we could do with this model? And then the key insight here is that there is an importance in timing. So this is the same 3D tumor model, the same drugs, the same dose, but at a different time. This could lead to a very different outcomes depending when we administer the drug. And yes, you can see that this question would be extremely difficult to understand, to answer experimentally. There is simply too many combinatorial problems that we would need to do in a real life in order to find the best administration scheme. So yeah, again, this is not a future vision. This is something that we can already done today. Yes, the second example of the human digital twins is the cancer cell invasion. Here we simulate not only cell proliferation, but also extracellular matrix degradation. And this model was able to reproduce both, it was able to reproduce the collective invasion patterns. So in this case, this small digital twin was not focused on the drug response, but on the understanding of the physical and mechanical way of the drug of the cancer invasion. Yes. And this is the third example where which just shows the huge magnitudes of scale that you need to support in order to have at least a partial digital twin. So here we combined actually two different tools. One of them is PhysiCell, which I'll talk about a little more, and a second one is Alia. So on the left, there is this ruler, which is actually the time, and each tick means a different process, a different time scale was triggered. And everything on the top is the PhysiCell, is the thing that I already showed, but everything on the bottom is something that we added more to this simulation, and that would be the dynamics of how we breathe the air, and also the fluid dynamics of the blood that is circulated in our body. So yeah, if you want to have a whole digital twin, you need to think about combining multiple different models and tools which act on a completely different scale, spanning from fractions of seconds to multiple minutes. Okay, and finally, I want to mention the European Virtual Twin Manifesto. So there are many bodies that are trying to answer this question of digital twin, and the European Commission is articulating that yeah, this is a reasonable thing to do, and we should yeah, we should aim to create an ecosystem of interoperable and validated digital twins that would be addressing some specific clinical questions. Okay, so this was a 20-minute long intro, and now let's move to the real part of the presentation, which is PhysiCell. So PhysiCell is agent-based modeling framework. And what does it actually mean? Typically, you have two main things. One thing is the domain. And inside this domain, you have something called agents. And in our case, the agents are cells. Each cell is represented by a very simple shape. It's just a sphere or a circle, if we are in 2 dimensions. And each agent or each cell has some properties, such as size, by that I mean the diameter or the volume, the position in the environment or the domain, and some internal state. Further, it can have some intracellular models, that alter the behavior of the cell. And yeah, there does not need to be just one cell in the domain. You can have specific composition of the cells, such as you can imagine a 2D monolayer, such as Petri dish, or more complex shapes, such as spheroid or dactyl shape of cells. So I I said multiple times term multiscale. So what does it actually mean? So PhysiCell is composed of specific scales or specific processes. So we typically draw it like this, with this ruler, where it is not centimeters but seconds. And again, each tick means one specific process is happening in time. And we distinguish 4 different processes. The diffusion process, which handles the diffusion of some substrate, which, as you can see, happens the most frequently, each 0.01 second, which is almost, well, around, sorry, 0.01 minute, which is around half a second. Then, a little less frequently, happens the cell movement. So this governs how the cell moves with regards to each other. This happens every 6 seconds. Then we have the phenotype or cell processes, which happen every 6 minutes. And then we have some regulation, some intracellular processes, for example, the Boolean network or flux balance analysis, which can happen up to every 10 minutes. So these are the different, we call them scales because they're happening in different points in time, in different time scales. And yeah, indeed, each of these processes is handled by a different, or governed by a different equation. And let me talk a little bit in a detail about each and every scale here. So the diffusion, perhaps the most important one. So the most important thing you need to understand from this slide is this short video on the bottom right. This is what the diffusion is supposed to do. Imagine that you have some substrate in your domain, such as an oxygen or a glucose, and yeah, this substrate is not evenly distributed in the domain. Imagine that on one part of the domain you have more substrate than in another one, and the diffusion is responsible for diffusing this substrate evenly or depending on your parameters of how fast the substrate should diffuse. Yeah, so this is happening every half a second, as I mentioned. So this is the time discretization, but the diffusion also have a thing called space discretization, which is in units called voxels. Voxel is just a name for a 3D pixel, a volumetric pixel. And you can imagine a voxel as a pixelation of your domain. So yeah, here I have a shape here that the domain is actually composed of these little cubes called voxels. And the more voxels you have, the more precise is your diffusion. So imagine if you had just 4 of the voxels, your substrates, well, each voxel holds just one value of that specific substrate. So if you have just 4 voxels in the whole domain, it would mean that you would have very pixelated domain, very pixelated substrate distribution, and the precision would be very, very small, very low. Yeah, I wanted to talk a little more about this because I think that the equation underneath is pretty interesting. So actually the diffusion is governed by solving thousands of simple linear equations. So I believe that you are aware about linear equations. So you have a matrix then you have a vector of unknowns and a vector of the right-hand sides. And the problem of solving linear equation is that you are trying to find this vector, so that the equation is correct. And the funny thing here is that the way how this works in the diffusion is that this matrix has just its main 3 diagonals being non-zero, and these values in this diagonal is actually the parameters how you want your diffusion to work. So there would be a diffusion coefficient there, meaning how fast your substrate diffuse, a decay rate, which means how fast the substrate decays in time, and the discretized time and discretized space. Well, so this is a constant, and then you, as a vector of results, you just put a row of voxels there. Then you solve the equation, and in the vector of unknowns is actually the next time step, this row diffused in one time step later. So it's actually a pretty straightforward or simple to imagine. But we actually have multiple of these rows, right? So yeah, we actually need to solve this linear system of equations for each and every row, but not just in one dimension, right? We need to do it also for each column and for the rows in the third dimension. So yeah, we need to solve thousands of systems of linear equations to move our diffusion in just one time step. And yeah, there are actually many other ways how to solve the diffusion. And PhysiCell is not the only tool that, not only one agent-based modeling tool, there are many others. I can name TSIM, CompuCell 3D, Computex, Yala, and each of them are solving diffusion in a different way. Typically, we distinguish between so-called time-implicit and time-explicit diffusion schemes. PhysiCell uses the time-implicit one, which means that it is unconditionally stable. What can actually happen in these diffusion schemes that if you do not have unconditionally stable diffusion models, it can happen that if you set your time step to be big enough, or voxel to be big enough, the simulation may explode. The error is unbounded, while in implicit time schemes the error is always bounded by some function. So typically you trade computational complexity for some bounded error. So in this case, our error is bounded, but you need to solve many linear equations, which in other diffusion models, the number of operations would be lower, but you would need to pay attention to how you set up your parameters of the diffusion so that your simulation does not explode. Okay, the next step is the mechanics time step. And here, as I said, the cells are represented as simple shapes. And the mechanics is responsible for how the cells move with respect to each other. So imagine that if the cells are almost close together, there is some force of attraction between them, so they want to come closer together. But also if they are overlapping, we do not want them to overlap too much, so they are repulsing each other. So these are the pairwise interactions that you need to do in order for the cells to move naturally, right? There are also other different movements, such as motility. And in this movement, this actually interacts with the previous step, the diffusion one, where you can specify a cell to move towards some substrate concentration, or not towards, but from some substrate concentration, which we call motility. And finally, you can have also something like a spring-like attachment. So you can attach two cells together, and you can imagine them to be attached by strings. So they will not be able to separate themselves away too much. This is useful for some simulations. Okay, the next one is the phenotype time step, or the cell growth or cell processes. In PhysiCell, we are supporting various cell cycles, and this time step actually is the one where you can move from one cell cycle state to another, which in return governs whether your cell will grow or duplicate or migrate or whatever you want. We can say that this is the intelligent part of the simulation, because here you can set some specific rules to the cell, such as start to migrate or stop to migrate, or start attacking another cell, or eating another cell, or anything else. And finally, here is the intracellular time step. This is a time step where you can put your own logic if you want to have some more complex logic over the cell cycle. So one of the most important use cases here would be the Boolean network. So you set up your signaling pathways such that your Boolean network will influence in which cell cycle your cell is, for example. And yeah, depending on some influences from the external environment, your intracellular processes can alter the cell behavior. And here it is, everything all together. I'm mentioning here PhysiBoss, which is just PhysiCell plus Boolean intracellular process, Boolean network intracellular process. And this is actually the drug example that I showed before. So yeah, here we have some Boolean network that is set in a way that if there is enough substrate, and by substrate I mean a drug concentration in the environment, the cells would stop growing, and when there is not enough of this substrate, the cell would continue to grow. It is simple as that. Yeah, in the videos you can see how the cells grow. The substrate here is not visible, but yeah, it is there. Okay, so maybe before I continue to another part, are there any questions so far? Okay, so let me continue to this quite interesting concept, which is the research that we've been doing pretty recently, and this is the more fine-grained cell shapes. So far, I've been talking about cell-based models and that they can be somewhat powerful, but interestingly, these models can have very simple geometry, right? Each cell is represented just by a simple sphere. But despite their usefulness, these models can often fall short in some scenarios where the cell shape has importance in how you do the simulation. So you can think about processes like cell migration, deformation in confinement, polarity-dependent division. These things would definitely be possible to simulate in center-based models where your cell is just a sphere, but it would not give a precise and accurate results. So yeah, we need more detailed approach to faithfully replicate these dynamics. And this is where the shape-based modeling becomes essential. So if we had a way to add a shape to the cell, add a shape of a cell to the simulation, it would allow us to capture more realistic mechanical interactions and simulate maybe even some intracellular structures. And yeah, the question is how to actually represent the shape computationally. And there are actually various ways how to do it. Maybe the simplest one would be to represent cell as a deformable sphere, which you can see on the left. And yeah, this would have a very small computational overhead. It is almost the same as the sphere, but it would not bring a big benefit. A more clever way would be to represent the cell not as a simple circle, but as a cluster of smaller circles called agents. And this would allow us to simulate completely amorphous shapes of a cell. And orthogonally to this, a pretty exotic approach would be to also represent shell shapes as springs coming through the center of a cell, where each spring would have a different length. So it would be equivalent to having different amorphous shapes. In our case, we chose to go with the second scenario, the subcellular agents, because we think that it is the most natural step from the center-based, where you have just one sphere as a cell. Okay, and so now we have a cell which is a cluster of agents, and these agents are simple spheres. So we need to enable more nuanced interaction between these elements, and we do this by so-called potential functions. There are actually various of potential functions. Maybe you heard about Morse potential, Lennard-Jones, Kelvin-Voigt. Typically, they are all trying to do the same thing. A potential is a function between two agents, and it governs how these agents interact with each other. So this is actually a function of the Morse potential. It is not really important. The more important is the graph here. So on the x-axis, you have actually the distance between the centers of two agents. And on the y-axis, you have potential energy. So naturally, on the bottom of the graph, where the potential energy is the lowest, the cells are in rest. And typically, you want cells to be at rest when they are exactly touching each other. Then there is you need to put some more energy into the system in order for cells to either split or try to overlap. And well, on the right, to this well, you have the higher distance between cells, which means that, as you can see, there is still some potential energy. So as they are just close to each other, they will try to attract. And on the left, we have quite a big step up in the potential energy, which means that if the cells will be overlapping, there will be quite a big bump in order to overcome if we want them to overlap more and more. So this actually is prevention of overlap of agents, which we actually do not want, right? Yeah, so this is just Morse, and yeah, every other variant is just modifying or tuning this function in a different way.